Hello everybody, I'm Dr. Imad Ibrahim. Today we'll talk about practical electrophoresis or gel electrophoresis in brief. What is electrophoresis? It is a device that separates macromolecule like DNA, RNA, protein according to their molecular weight, اللي هو size, and charge, positive or negative or neutral, and a shape اللي هي conformation shape, الشكل الفراغي. It is a device separate the protein, nucleic acid, and DNA, RNA according to their charge and molecular weight and shape. Here we talk about gel electrophoresis. Gel electrophoresis, mainly we use it for DNA separation analysis or RNA, and sometimes in hemoglobin, in acidic citrate agar hemoglobin electrophoresis in acidic pH. We have a tank. The component of electrophoresis is a tank. Contain positive electrode and negative electrode and a supporting media which is gel. عشان كده we called it gel electrophoresis according to supporting media. And this gel contain wells, some spaces to put the sample. When we make the gel we put a comb, بنخط مشط عشان يعمل فتحات، يعمل لنا spaces اللي هي spaces to put the شنو to load the sample. This is wells to load the sample. Then we put buffer. What is the function of the buffer? The buffer will resist the change in pH. لأنه pH is very crucial في الanalysis of protein. لأنه الcharge تحت البروتين, charge of protein will be determined by the pH. If you put acidic pH or basic pH, the protein will change its charge according to the pH. However, the DNA and RNA is fixed, are negatively charged. DNA and RNA, due to phosphate backbone, they are always always negatively charged. So they will migrate towards the positive. However, the protein, according to the pH, we always use buffer. The function of the buffer to resist change in pH, to conduct the electricity, to act as a conductant medium عشان يوصل الكهرباء between الجل والpositive and negative electrode, which permit the sample to be influenced by the electricity to migrate either towards the positive or towards the negative. So this is the importantness of the buffer. So we put buffer solution. Then we connect. Then we can load the sample. Here we talk about loading of DNA sample. The DNA will be mixed with a loading dye. It's the blue, phenyl blue for example. It is a blue color to show you that you are to show the sample inside the well, to know which is the empty well and which is the loaded well. طيب. A loading dye, it also contains density agent like glycerol. Why? To make the sample loading down inside the well. لأنه if there is no density agent, the sample can be washed off in the solution. تطلع من الويل تخش السوليوشن. لكن we mix the DNA sample with tracking dye to see the sample when it migrate, and with the density agent to maintain the sample inside the well. After loading the sample in the wells, many samples according to the number of the well, then we connect the sample loading inside the well, under the buffer. Then we connect a power supply which supply us voltage and milliampere, and we adjust to the suitable volt and milliampere and put it for a suitable period of time, 1 hour or 2 hours. Then DNA will migrate towards the positive. because DNA is always negatively charged. It will migrate towards a positive no. This is what happened. This is the gel from above, and this is the well, and we load the DNA sample which mix with loading dye in the well, as we can see. We load the other well, so we put positive electrode and negative electrode. This DNA will migrate towards a positive. Then the DNA will be separated from each other. Each fragment of the DNA will be separated, and we have DNA marker to measure the size of this DNA fragment. The large will be close to the origin, and the small will be away from the origin towards the positive electrode. Because here in the DNA, all DNA are negatively charged, so the separation will be according to the size. Small fragment will migrate in the front, and large fragment will be at the back. This is real picture of the gel visualized under UV light, because we will put in the gel iridium bromide. Iridium bromide is a fluorescent dye. When we make the gel, we put iridium bromide, so when we load the DNA and it separate, iridium bromide will intercalate the DNA. لأنه DNA will not be visible. We cannot see the DNA. When we stain with iridium bromide and we put the gel under UV light, the DNA will be visible like this. It will be illuminated. الDNA بيكون ظاهر لأنه انصبغ لي intercalated with الإيسيديوم برومايد، and الإيسيديوم برومايد under the UV give شنو will be eliminated. فبيظهر لنا شايفين هنا on the left this is DNA marker, and on the right 1 and 2 these are samples separated to different fragment of DNA with different lengths according to the size. This is the end of this lecture and of this session tutorial about DNA electrophoresis or gel electrophoresis. Thank you very much for your listening. Thank you.